present all around us, even inside our bodies. And they encompass a wide range of the organisms, including viruses, bacteria, fungi, algae, and protozoa. Microorganisms can be useful for food and beverage industries, medicines and vaccines, agriculture, and cleaning the environment. Microorganisms can be also harmful for causing disease and spoilage of food. Microorganisms spoil food. Sometimes the food may get spoiled due to varieties of reasons like not keeping it covered, improper cooking, or not preserving the food properly, and the change in the temperature of the refrigerator and food due to power fluctuations. Now, I would like to call my friend Simran to continue the presentation.
Now I would like to explain about food preservation. Food preservation is the process of treating and handling food to slow down or stop spoilage, thereby extending its shelf life and preventing loss of quality, nutrition value, and edibility. There are many ways to preserve food. Preservation by dehydration. Dehydration preservation, also known as drying, is a method of food preservation that removes inactive microbial growth and extends shelf life. This process lowers water activity of food, making it unsuitable for bacteria, yeast, mold to thrive. Preservation by chemical method. Chemical preservation uses substrate to inhibit microbial growth and extend shelf life. Extend food shelf life. This method involves adding preservative like salt, sugar, vinegar, synthetic compound to inhibit spoilage and maintain food quality. Preservation by common salt. Common salt or sodium chloride is widely used food preservative primarily due to its ability to inhibit microbial growth by drawing out water and create a hypertonic environment which is unfavorable for many microorganisms. Now I will call my friend Fiona for continue the presentation. Now let's tell you about preservation by sugar. Sugar acts as preservative by reducing the amount of available water in food, which inhibits the growth of microorganisms like bacteria and fungi that cause spoilage, creating a high pressure that dehydrates and kill microorganisms called spoilage. Now I'll explain you about preservation by oil and vinegar. Oil and vinegar are used to preserve food by creating an environment that inhibits the growth of microorganisms. The acidic nature of vinegar and anaerobic environment that created by oil together prevent bacteria, yeast and mold from spoiling food. Thus, it's extending its shelf life. Now, I would like to call Darshi for continuous speech.
Now, I will explain about pasteurization. Pasteurization is a process of heating liquid and some food at specific temperature for a set time to kill bacteria and exchange shelf life. Means this is the process of food which is heated first and it's cooled down quickly to kill the bacteria. This is the diagram of pasteurization with example of milk. Fresh milk from cow enters to heating section. There it heats around 70 degrees Celsius for 15 to 30 seconds. Then it enters to cooling section. There it cools around 0 to 10 degrees Celsius to prevent the growth of bacteria. Then this means stored in sterilizer water or in pouches. Now I will explain about preservation by cooling or freezing. Cooling or freezing are effective methods for food preservation, slowing down microbial growth and enzyme activity to exchange shelf life. Cooling, typically using refrigeration at 0 to 4 Celsius, it inhibits bacterial growth and exchange shelf life. Now I will like to call my friend Shreya to continue the presentation.
My topic is preservation by canning or vacuum packing. Canning involves heating food in sealed containers to this canning. Here we have canned some jam, pickle and ketchup. Here you can see that we are adding a milk. Here you can see that we have added here milk to boil it for 15 to 20 seconds. That how many bacteria are present in the milk, it would be killed. Here you can see that we have preserved many vegetables, fruits and some food that we can use later. Now I would like to call to Darshan to explain more.
Now, in this model, I will explain. In this model, I will explain about foods. This part is about meat and alternatives. In meat and alternatives, there is non-veg like including chickens, chapati, fish, etc. In this part, there is about milk products. In milk products, including like ice creams, milk, ghee, etc. In this part, there is fruits and vegetables, including like tomato, apple, mango, potato, carrot, etc. In this part, there are grains and pulses. In grains and pulses, including bread, rice, nuts, then pea, etc. Now, I will like to call Manvi to explain our model.
These are the types of organisms which are present in our food and even in our body. This is amoeba, this is paramecium, bacteria, Euglena, viruses, Spirogyra, bread mold and Vibrio cholerae. And at last, I would end my speech with a quote